Hi. While others discuss burning issues of a day on YouTube scoring hundreds of thousands views, I continue my narrow-minded stories about big data and visualization stuff, hoping for better times in the future. Or maybe one day I will switch to a topic like should I shower every day, or how to spot an idiot. No, not really. Previously I showed you how you can convert BAM files, which are shown here in IGV viewer, into the bigwig files and then take average of bigwigs, and then instead of such a busy figure you will have only two tracks and have lots of space for other tracks. But the story was not complete yet, because next to BAM files you have a bed files, and loading individual bed files for every sample sounds not optimal. The example I show you here is available in NCBA Geo under this number, and faster files are available in SRA. Essentially, 7 samples are split on 2 cell types. 1, 2, 3 is neurons, and 4, 5, 6, 7 are iMOCs. The role of the bed files is to integrate peaks in your genomic landscape. If you load them individually per each sample, then you will get dizzy pattern, which obviously requires cleaning and simplification. And whereas in case of BAM files you use the deep tools, in case of bed files you will switch to Bedtools, and then they will help you of making average files. However, speaking of average, don't take it literally. More likely we will want to have a consistent peaks. Within the same cell type, these two peaks are consistent, they reproduce at least twice, and here again this reproduces twice, but this peak is not consistent and this is neither, because they appear only once and then we probably can ignore them. Let's check what options we have in a Bedtools kit. Here is the full list of functions, out of which you have to choose, and what I personally use are those two. First, you apply multi-inter for bed files in each group in separate, and it will mark all locations where peaks overlap. The disadvantage is that your original peaks, those indicated by lines, will be fragmented, and therefore in the second step you apply merge, which will merge peaks in a neighborhood overlapping or a little bit on a distance. That's what I will show you here. And do not take it as the one and only trick. There are many more ways of doing more or less the same averaging. That's only one solution out of many other possible. If you never used Bedtools before, then of course you have to install it. I used it before mostly in Linux, but why not doing it in my Mac OS? And for Mac OS, the easiest way is to use a homebrew. Just type brew install Bedtools, and then the rest will work by itself. Yeah, and now it is responding. When I worked in Linux, I installed Bedtools with apt-get and used the Python script. In macOS, Python does not connect well to the terminal, and therefore I used RScript instead. It's a bit strange feeling to use R for such a simple task, but it works. So first we set a working directory where your bed files are, then select narrow peaks, then check what for files are here, and then according to description of this particular data set, I define this first group as neurons and the last as IMOPs. That's the lines where I make them. Pay attention, it's not a list, it's a long string. And then I say let's do it for neurons first. Then I can call make directory average using system command. This is essential function to communicate with the command line, and it will say file exists or directory exists already, no need to call again. Okay, and then because we took neurons, our output will be neurons multi, because we use multi-inter. Here we generate a line, all according to instruction. Then we can also ask what my line looks like. It looks like this: Bedtools multi-inter -i, then the list of files, then it say direct to the average neurons multi TSV, so the result will be saved into this file. We call it, it works. Then we go to this subdirectory and read the file, and then have a look at it, and this is quite a simple headless file where these two are two versions of the same. How many of individual tracks overlap in each particular bin, and then we say we can also count them and say most of them do not overlap, then 2 overlaps in a 5000 plus, 3 overlaps in 1000, and we say selected is a file where v4 column is more or equal to. What you have to select is the first 3 columns and say first 3 columns, 1, 2, and 3, so chromosome start and end. The rest is not relevant for a bed file, neither for visualization. Select and then write the truncated filtered table back to the file called filtered. We did it, and then we go to this subdirectory list files, and then it is a little more because I did it already. So let's pretend we don't know. And then we generate another line where we will merge all these selected peaks, and then we also can call my line to see how it looks. Bedtools merge -i, neurons multi-filter TSV, now it is only one file. -d50, it means that if the distance between two peaks is less than 50, call it as 1. And then the output will be named neurons multi average bed. It's a little bit too many multi. I would say this is enough. And then call system, and then system is done. And in principle, you can make complete bed file in a script, but I would suggest to do it in a Excel sheet or OpenOffice, like this one, for instance. You open the file you want. So that's the structure. If you don't like these unassigned chunks, you can remove them. Pay attention that there are no headers, no row names. What you have to add is a name, and the name will be neurons. And here is the orientation, which strand it is. In case of the chipsec data, there is no orientation of your peak, therefore it is a dot. Dot is actually problematic symbol in R. Sometimes it works, sometimes it isn't, because it's also part of the regular expressions where it means everything, if I remember well. And now what you have to do, you have to make those columns by copying neurons dot and then pasting through all the up to here, and then you say paste, and now you say save. I would say save as, save. Yes. It is important that the extension is bed, and we have an average bed only 3 columns, and average 2 bed is the same 3 plus neurons plus dot. Now we go to IGV. I preloaded already individual bed peaks. Now we open our generated average and average 2. And then you see a difference. So if average only 3 columns doesn't show anything, it's incomplete and therefore it's not taken by IGV. Average 2 has a neuron sign and neurons appears right here, and the directionality also helps to visualize it. That's how it works at the end. Knowing that, you can move to your source annotated file. This table is made by divbind protocol, where it documents all differential peaks found in this data. And again, select those columns, then say new, paste, and then you make bed file out of it. You have to delete header, otherwise again it will be rejected. Now here you can say all differential, so these peaks are differential, and here the star is a wrong symbol, actually made by R, and then copy paste, yes, and then save as, CSV, differential, and then bed, if it will work in one go, save, CSV, and then field delimiter should be tab, string, okay, I don't care, probably, okay. It's still not okay because it's adding CSV. So remove CSV, use bed, check inside, chromosome start and name orientation, and now it is ready to be loaded. Differential, open. And so we know that this peak is differentially expressed according to a divbind, and it has a name and you cannot be confused. And at last you can also add average files, mobs neurons, so that's the things. And of course I have to load imobs as well. And after this, there is no point of keeping empty and individual tracks. Remove. That's the minimum you're supposed to show about BAM and BET files. Of course, I mean this particular case where we have a chipsec experiment comparing two groups of cells. One is 3 samples, other 4. Comparison in simple neurons to IMOPs, and differential peaks are detected using DiffBind. In this case, it is enough to show average for IMOPs, average for neurons, whether it is differential peak, it is. How it looks in neurons, the peak is here. In IMOPs, it's shifted to the left. That's a curious case where DiffBind claims a difference, which is actually difference in position, even though those peaks overlap considerably. We can check other locations. Here, for instance, it says that IMOPs shows peak and neurons they don't. So this IMOPs peak is differential and present only in IMOPs, and so on, so on, so on. That's more or less the end result of averaging of BET and BAM files.